Welcome to the latest episode of Star Cells and God. My name is Fuz Rana. I'm a biochemist and a Christian apologist, and I'm joined in studio today by Dr. Hugh Ross, who is an astronomer, a Christian apologist, and a founder of Reasons to Believe. This is the organization, by the way, that sponsors this podcast. Today we're going to be talking about two discoveries that provide evidence for the Christian faith. Hugh's going to be talking about the latest claims about scientists discovering a habitable planet and what that means for Christianity. And I'm going to be talking about some work by German researchers that add to the case that a creator must have played a role in the origin and the design of life. You don't want to miss it. Well, Hugh, good to see you again and great to be in studio with you. You've got a really interesting discovery that I am familiar with because I saw headlines all over the place.
Yeah, it's all over the web. So
I'm glad that you're going to be here to unpack that discovery for us. So why don't you go ahead and take it away?
Well, the reason it got so much web attention is that these astronomers were not just claiming that they had found a habitable planet. I mean, there's lots of planets they've been finding where liquid water could conceivably exist on the surface. They're actually claiming they'd found life on another planet. So that explains why you had all that buzz on the internet. And it's not the first time this team has made that discovery. They made it almost 2 years ago, where they found a particular molecule in the atmosphere of a planet, and it's dimethyl sulfide. And the interesting thing about that molecule is that on Earth, the only source for dimethyl sulfide is emission from marine phytoplankton. And so they're saying, hey, on Earth, it comes from no other source but life, and therefore the fact that we're seeing it on a planet called K2-18b means there must be life on that planet. Yeah.
So how certain are they that they actually have a spectroscopic signature for
dimethyl sulfide?
Yeah, for DMS.
Well, 2 years ago, they said, hey, we think we see dimethyl sulfide, but the significance was between 1 and 2 sigma, which isn't going to impress either a chemist or an astronomer. You might say that's a possible discovery, but it's certainly not very probable. What they did is they got time on the James Webb Space Telescope to get a much better measurement of the spectral line. And this time they came back and said, we have a 3 sigma detection, which they basically communicated means that they were 99.7% certain that they had found dimethyl sulfide in the atmosphere of K2-18b. And that's what got all the attention on the web. Wow, 99.7% certainty. But the problem is that these popular articles really weren't communicating the significance of 99.7. I mean, if you're looking at, say, 1,000 spurious sources, one of them is going to come up at 99.9% certainty. Yeah. And so, like, if you're doing a broad spectrum survey, in fact, it reminds me of the days I did radio astronomy, where I would see noise, random noise, and if you look at enough random noise, you're going to get quite a few signals that are above 3 sigma, which is why in science, you don't really have a discovery unless it's 5 sigma, 5 times the standard deviation, which basically means you're 99.999% certain that it's real. That's kind of the standard to claim a discovery. Now, if you read the paper's right here, constraints on DMS and DMSS in the atmosphere of K2-18b from the James Webb Space Telescope. In the paper, they're quite clear, hey, this is not up to the standard of a discovery that would win you the Nobel Prize. Right. But hey, it's a way better than what we had 2 years ago. Right. So that was kind of what they said in the paper. And they're basically, it was quite a long paper, but they end the paper by saying, given that we're now up to 3 standard deviations, we think it's worthwhile getting a whole lot more time on the James Webb Space Telescope. So they're basically pitching for more telescope time. And time on the James Webb Space Telescope comes at a premium.
Yeah.
Everybody's trying to get time on the James Webb Space Telescope. So, this 25-page paper is basically saying, hey, look what we found.
Right.
We think we deserve more time so we can get it up to 5 sigma.
Yeah.
So, but, in the popular press, they're saying, wow, these people have really found life on this planet. And so, and we did a podcast on this, a couple of years ago. And so people can go back into Star Cells and God and see what we reported on. Pat, I think you were on the show with me when we talked about it. So this is kind of round 2. And what we said 2 years ago is, number one, there's good reasons to believe that K2-18b is not a candidate to host life. I mean, yes, it may be in the water habitable zone, but they ignored all the other habitable zones that are requirements for life, and it's orbiting the wrong star. It's orbiting a red dwarf star, an M3 red dwarf star. And the problem with red dwarf stars is that they pour out a lot of flares, and these big, bright flares are catastrophic for life. Moreover, because the star is so dim, the planet has to orbit a lot closer to the star. So this planet is orbiting only about half the distance, less than half the distance that Mercury orbits the sun.
Oh, wow.
So it's orbiting very close. And it may not be tidally locked like the moon is to the Earth, where only one side faces the Earth. But we do know for certain that the planet is spending a minimum of 16 days per revolution. The revolution period is 33 days. We know that it's spending at least 16 days where one side faces the star. And that means you're not going to have life. It's going to be really hot on that side, and it's going to be freezing cold on the other. This is not a candidate for life. So, but because it's relatively close to us, the James Webb Space Telescope can get very good quality spectra, which is why I think they chose. And it is in a zone where liquid water could conceivably exist, but only if you're talking about a planet that's rotating like our planet. This planet isn't doing that. It's receiving so much tidal force from the host star that it has a very long rotation period.
Yeah.
So for lots of reasons, it's not a candidate. In other words, I would argue it shouldn't have even gotten any time on the James Webb Space Telescope, at least for not that mission.
Yeah.
However,
I mean, so how would you respond to the argument that where somebody might say, well, maybe we have too narrow a set of requirements for what would be the conditions for life. Perhaps there could be life forms that might be able to tolerate a much broader range of conditions or more extreme conditions than what we see here on Earth. So, so that your assessment, while valid for life on Earth, maybe isn't valid for life as we don't know it.
I remember a chapter you wrote in our book, Origins of Life, where you made the point that extremophiles are not a sign that you can have life, because you need mesophiles in order to sustain the extremophiles.
Well, an extremophilic origin of life is just even that much harder than a mesophilic origin of life.
Exactly. So the existence of extremophiles is, it's not a good argument for the origin of life. Although it's interesting, Fuzz, just today I got the latest issue of Astrobiology, where a group of biochemists like yourself were saying, hey, we can make simple lipids within sulfuric acid. So they're basically saying maybe life could exist in the atmosphere of Venus. There's a lot of sulfuric acid there, but they're actually arguing that could be a suitable solvent for life. But they went to a lot of effort to make these lipids, and they're quite simple. And we're only talking lipids, and who knows how quickly those will break down in sulfuric acid. There's a consensus in astrobiology that life needs to be based on a water solvent. Sulfuric acid, you're a biochemist. You know more about this than I do. Water really is the only game in town. So
and it needs to be liquid water. You can't have
water,
a gaseous environment because there's just not sufficient molecular interactions in a gas phase for life to really exist.
Well, let me go back to the dimethyl sulfide, because this is right up your alley. And several papers have been published literally before this paper came out, one just after this paper came out, basically calling into question that dimethyl sulfide is a biomarker. So it's a biomarker on Earth. I mean, the only way you can get by dimethyl sulfide on Earth is through these marine phytoplankton. But 3 papers got published. One that got published before this paper came out, basically making the point, hey, we've already discovered dimethyl sulfide in the coma, that is a gas tail that comes out of the comet 67p. That was detected a couple of months ago. And I think I got the paper here. Yeah, here it is. Interestingly, published in the same journal that they published, but a little bit earlier, saying evidence for abiotic dimethyl sulfide in cometary matter. And the comet was 67p. And they're making the point in the paper, there's no way that that gas tail coming out of the comet would be a habitable region. So we know that it has to be abiotic. So, and then also about the same time, a group of biochemists actually found several abiotic pathways to make dimethyl sulfide in the lab. So both of these papers concluded we need to strike dimethyl sulfide off the list of being an unmistakable biomarker. You could say it's a possible biomarker, but it's not a definitive biomarker.
Right. Well, and you know, when you're dealing with different planets that have different geologies and geochemistries, what is true here on Earth in terms of production, like a compound like DMS, may not necessarily be true elsewhere. So, you know, it's almost like the opposite of the argument that people make for life being found in places that, you know, we would naturally think that life isn't going to be possible. It's almost
That's
the counter
exactly
argument,
the
right?
point that the biochemists made when they were making dimethyl sulfide in the lab. They say, hey, on Earth, you got relatively warm conditions. But if you got a cold environment, you can make dimethyl sulfide abiotically. And so, you know, the tail of this comet was a cold environment, and it had a lot of, you know, rich carbonaceous molecules there. So there are plenty of abiotic pathways to make it. So on Earth, it doesn't happen, but if you got colder temperatures, it can. And then just after the paper that got published where they claim, hey, we think we found signs of life on K2-18b, this paper got published where they said, we've actually detected dimethyl sulfide in a giant interstellar molecular cloud at the center of our galaxy. Now, there's a supermassive black hole at the center of our galaxy. This is clearly not a place where life can exist. Moreover, interstellar molecular clouds are not a candidate for hosting life. But what's interesting about these very dense giant molecular clouds is you get a whole array of quite complex biomolecules. I mean, to date, I think there's been 145 that have been discovered. And the one that where they made the most discoveries is a giant, dense molecular cloud at the center of our galaxy. And the reason for that is a really big cloud. It's illuminated brightly by all the stars at the center of our galaxy, so it's an easy place to detect these lines. And it's filled with carbon, nitrogen, and oxygen. So you have this very dense cloud of carbon, nitrogen. Obviously, it's a great environment for making the building blocks of biomolecules. And so just a few days ago, this paper got published where they said, we have found dimethyl sulfide in the interstellar molecular cloud at the center of our galaxy. Clearly, it's abiotic. So we have 3 papers published in the last 2 months, basically making the point, there's multiple different environments and multiple different ways you can make dimethyl sulfide abiotically. And so the claim in this paper, hey, we think we found life, is based on the assumption that dimethyl sulfide was an unambiguous biosignature. We now know that it is not. And I think the real kicker was a team of researchers that actually published a paper 2 months after the one in 2023, came out with a paper this time basically making the same critique, saying, hey, we're not sure this team has done a correct analysis of the spectral line. because what they did is they took the data that they published and analyzed it with what they claim a superior, more reliable method of analysis. So when we do that, the evidence for the spectral line disappeared. So they're even claiming, they're calling into question, did they really come up with a 3 sigma detection of dimethyl sulfide? They're basically saying, hey, when we tried to repeat what they did, using what we think is a superior analysis of the spectral data, we don't see this significance. So I think the end result is I'm not sure they're going to get their time on the James Webb Space Telescope. And the team that made the reanalysis says, look, even if their analysis is, quote, better than ours, and we don't think it is, what we notice is the James Webb Space Telescope has already stressed to detect that line at a 3 sigma level. There's no way you're going to get a 5 sigma detection, because even when you look at the noise around the 3 sigma detection, this is at the very limit of what the James Webb Space Telescope can do. And so dedicating more time is not a good thing, because this is what they published is basically telling us this is the limit of what the James Webb Space Telescope can do. And they complimented the researchers saying discovering that limit is actually a useful scientific advance. Now we know what we can do with the James Webb, what we can't do. So this is actually going to make radio astronomy, optical astronomy, much more efficient.
Right.
So they said, we're not dissing you entirely. They did call into question some of what they had published, but they said, nevertheless, we think you've made a valuable contribution because you've shown us the limits of what the James Webb Space Telescope can do. But this kind of reminds me of my friends who are involved in the search for extraterrestrial intelligent life. I mean, hundreds of hours, thousands of hours have been dedicated to the search. And it's like, this is valuable telescope time that's being dedicated to this. Shouldn't we take some caution to ensure that we got, reasonable candidates? And I've been arguing this for the past 50 years, is that instead of trying to collect a database of what we think are planets that could conceivably host intelligent life, we should be looking at the stars. Do the stars have the characteristics that are needed for a planet orbiting that star to host advanced life? And and the whole point is it's a whole lot easier for us astronomers to measure the characteristics of stars than the characteristics of planets. And so if the stellar characteristics are such that we know there's no way that there can be a planet orbiting that system to host advanced life, it's like, let's not dedicate valuable telescope time to searching for these signals that we already know because of the star they're not a candidate. I think that analogy applies to K2-18b, is that we can look at its host star and realize there's no way any conceivable planet orbiting that star could possibly host advanced life. The flaring activity, I mean, what we know here on Earth is it takes a long time to go from simple life to advanced life. Even from the perspective of saying, hey, God's involved in this. Of course, all these papers are written from a non-theistic perspective. But I would argue that what we see in, say, in planet Earth is God performing miracles as aggressively as possible to bring advanced life on planet Earth as quickly as possible. But we're still looking at 3.8 billion years. So I think God could have done it more slowly, but I don't see how he could have done it more rapidly without altering the laws of physics. So that means if you've got a star that's pouring out these frequent deadly flares, and we're looking at the length of time it's going to take to go from bacterial life to advanced life, it's like, no, K2-18 is not a candidate to host a planet where you've got life existing. Even primitive bacterial life would not be a candidate. I think the most frustrating thing to me as an astronomer, Fuz, is all this research on astrobiology is focusing on a single habitable requirement.
Water.
Is there liquid water on there? And it's like, I've been publishing articles and books for years making the point, hey, we now know that there's 15 planetary habitable zones. And the liquid water habitable zone is the most generous of the 15. The other 14 are far more restrictive than the liquid water habitable zone. And life, even the most primitive life form, it needs to exist in the majority of those 15 planetary habitable zones. And even if you pick up the one that's been best studied, the ultraviolet habitable zone, what we know about this star is that none of its planets can simultaneously exist in both the liquid water habitable zone, the ultraviolet habitable zone. And hey, for any red dwarf star, the problem is all of its rocky planets are going to be in what's called the electric field, atmospheric electric field habitable zone, something that happened to Venus. Because of how close it is to the sun, it has a relatively strong atmospheric electric field, which basically causes all of its surface water to disappear relatively quickly. That's going to be the case for any red dwarf star. So all of the planets in this liquid water habitable zone will suffer the same catastrophe that Venus has suffered. We're only looking at 3 of the 15 habitable zones. And in addition, you got what are called galactic habitable zones. And where I think this impacts the Christian faith, it does in 2 ways. Number one, all these different habitability requirements, 15 for the planet, another 2 that we know of for the galaxy, and then, hey, there's even outside of our galaxy cluster habitable zones that we need to take into account. It's like all this is telling us that the only way you can have life on a planet is if you've got a supernatural, super intelligent God that's ensuring that all these habitability requirements are met. And, when you look at planet Earth, our planet is simultaneously exists in all the known habitable zones. We seem to be discovering a new one every couple of years, so the list is growing. And I think it demonstrates the principle you see in Psalms and in Job. The more we learn about nature, the more evidence we uncover for the supernatural handiwork of God. But I think this research also gives support for Romans 1. We have all this evidence for all these distinct habitability requirements that point to the fact this isn't going to happen naturalistically. It's going to take supernatural intervention. But what do these researchers do? They engage in Romans 1, 18 to 22.
Right.
Self-imposed ignorance. They focus on the liquid water, which gives them the most optimistic point of view, and they ignore all the pessimistic issues.
Well, one of the things that I think is very interesting when you look at Romans 1, 18 through 25, is this idea that what happens is people tend to worship the creation, not the creator. And I see that with so many scientists who rightly stand in awe before the elegance and the beauty of the universe and the world that we live in and living systems, and they marvel at it. It's almost like a religious response to the creation, but they're worshiping the creation as opposed to the one who made it.
Right.
There's a book that I like to cite. It's written by a woman named Ursula Goodenough, a molecular biologist. It's called The Sacred Depths of Nature. It's a classic work in spirituality and science, but she calls herself a religious non-believer. And the book reads like a devotional, where she just marvels at creation and has almost a religious response to it, or a spiritual response to it, but she just still denies God's existence.
It reads a lot like the 6 creation psalms that we see in the Bible without the conclusions of those psalms. Look what God has done.
Yes.
That part's missing.
It is. Well, as I heard you, talk about kind of the implications of this discovery you're talking about for the Christian faith, I'm immediately drawn to Isaiah 45:18, where it declares that the Lord created the earth to be filled full of life. And when you look at Genesis 1 in the precise sequence of events that are described there, where each one is preparing the planet for life. And you see this in Psalm 104, where each day of creation is setting the stage for the next day and the next day, that there's clearly this idea of just a series of just right events that God is executing in order to make life possible on the earth and ultimately human life.
Well, I kind of see that in this research. Here are these researchers trying to find exo-life, but the more they research the possibility of life beyond our solar system, the more evidence they're uncovering of all the different factors that must be in place for life to possibly exist. Even though they may be ignoring it, it is showing up in their published papers, and it's actually accumulating the case that indeed you need all this design. I also see this research as settling a Christian debate that's been running on for 2,000 years. And what's been happening, for the past 2,000 years is two groups of Christian theologians. One group looks at the creation Psalms, especially Psalm 104, and says there has to be life beyond planet Earth. Because you look at these creation Psalms, it seems that God is exuberant and just passionate about creating life in the greatest possible abundance and diversity here on Earth. And they argue that exuberance is bound to be expressed elsewhere in the universe. Then there's another group of theologians who say, yeah, but when we look at the Gospels, notice how frequently Jesus refused to perform miracles. Herod wanted to perform a miracle. Pardon me, not Herod. Who was it that was? Oh, yeah, Pilate.
Yeah.
Pilate wanted to perform a miracle, and he said, I won't do it. When he fed the 5,000, they said, do it again. He says, hey, you missed the whole point of my performing the miracle. And these theologians are basically arguing God does perform creation miracles, but he's conservative in the way he performs his miracles. He restrains himself to performing the miracles that are necessary to achieve his purposes. And they argue, when you look at Scripture, does God really need more than one planet in this vast universe in which you have, an uncountable number of humans that are struggling with sin and in need of a redemption? And they basically argue God doesn't need a second one. He can do it all with just one. So they would argue that we're alone in the universe amongst physical, intelligent life beings. Whereas another group of theologians are saying, no, God enjoys creating so much, he's not going to limit himself. Astronomy is now jumping into the picture and saying, everywhere we look beyond planet Earth, we see conditions that look not only hostile, but extremely hostile to the existence of even primitive life. And so they're arguing it really looks like we're alone. And they're also discovering, hey, just to get one planet, we need a universe exactly the size of our universe. So it's not like the universe is a waste. Given the physics of the universe, you really do need 2 trillion galaxies out there, where each of them have, hundreds of billions of stars. So astronomy seems to be settling. That's what I find fascinating. Here are these unbelieving astrophysicists settling a debate that Christians have had for 2,000 years. I wouldn't say the debate is settled yet, but I think it's moving in that direction. So,
well, the point of the book, or at least one of the points of the book I wrote called Fit for a Purpose, is that terrestrial biochemistry is universal biochemistry. That the requirements that you need for life at a molecular level are uniquely fulfilled in the biomolecules we see in living systems. And those biomolecules have to function in a very specific set of environmental conditions, requiring liquid water, requiring carbon dioxide at just right levels, requiring molecular oxygen at just right levels, requiring phosphates. So you just can't have any environment out there that could support life. So, in response to this idea that maybe life could be extremophilic, nobody's been able to show how you could have alternate biochemistry in other environments and have living systems. So, in a sense, biochemistry is adding to that argument as well. So it'd be one thing if we saw environments where conceivably life could exist. Then you could say, well, if we detect life there, that could be another example of a creation miracle. But the fact that we're not even seeing places where life could exist seems to rule out this idea that God has created life else, throughout the universe.
Well, that's kind of what I see in the astrobiological literature, is that if we find conditions suitable for life, we're going to find life. It's going to be automatic.
Yeah.
And it's like, well, look at all the biochemistry experiments done here in the labs where they have the just right conditions,
Right.
and they're not able to pull it off. I mean, people equally as gifted as you, with a lot more funding than you.
More gifted and more and better funded.
Definitely better funding, no question about that. And not just one, but a whole team of them. And yet they really are only able to do the baby steps that are necessary for the origin of life. So the idea that suitable conditions will automatically produce life doesn't bear out in the biochemical experiments we see done in laboratories. But also, we astronomers are not finding suitable conditions elsewhere. We can find some of the necessary conditions, but nowhere near all of the necessary conditions for life to possibly exist. And that's my critique of this study. We already know that K2-18b possibly could have water. And I say possibly. We don't know for sure that is water. In fact, several of the papers that have come out have basically made the point, look, we know it doesn't have water. There's reasons why. For example, the planet is way bigger than the Earth. It's 8 times the mass of the Earth. So it's basically Neptune-like, not Earth-like. And when you've got a Neptune-like, that planet that massive, you've got a very thick atmosphere. We also know that its size, its diameter is 2.5 times the diameter of the Earth, which means it's got a low density, basically 2.6 grams per centimeter. And you don't have to be a biochemist to realize you've got a condition where the density is not adequate for life to possibly exist. So there's a lot of reasons why, hey, it's not an Earth-like planet. And it's so non-Earth-like that we know that it can't be a candidate for which biochemical life processes can operate. So, hey, you want to talk about genetics?
Yes, I do. But to kind of give us a breather for a minute, I don't know if you are much of a baseball fan or not. I know your wife, Kathy, is a huge...
she's a huge fan, yes.
Huge Dodgers fan. But I love baseball, and I'm a Reds fan. You cut me, I bleed red. So
but my wife, you cut her, she bleeds blue.
Yeah, I know that.
Anyway.
But this is my favorite time of the year. We're recording this towards the end of April, and so we're about 1 month into the MLB season. And this is still the time where hope burns eternal.
For all teams.
For all teams. As a Reds fan, usually by the end of May, heading into mid-June, I've given up on the season. But for right now, there's still some hope. But I also, not only love this time of the year because of, again, the season starting and getting underway, but it just is a reminder to me of the rich history of baseball that kind of gets revived, for me at least, every spring. And a number of years ago, I read a great book, and I've got a picture of the book for our audience. It was called I Was Right on Time, written by a baseball player named Buck O'Neill. And Buck O'Neill wrote this book as an autobiographical account of the time that he spent playing and managing in the Negro Leagues. And of course, there was sadly a time where African-American players weren't allowed to play Major League Baseball, so there was a Negro League. And Buck O'Neill was a fairly respected ball player that played first base and later became the manager for the Kansas City Monarchs before the league kind of folded. And it was during that time when he was a manager that Jackie Robinson made the transition from the Negro Leagues to the Dodgers, right, where that grand experiment where the color barrier was broken. And, of course, that led very quickly to other Negro League players transitioning to the major leagues. And as a result of that, the Negro League just essentially again folded. But somebody once asked Buck O'Neill, and he tells this story in this book, are you sad that you came a little bit too early? Because many people thought, as good of a player as he was, that if he was, if it was 10 years earlier where the color barrier was broken, he would have probably made it to the major leagues. And his response was, which I always find to be quite endearing and really very healthy, was, no, I was right on time. Because I got to play in the Negro Leagues when the best players in baseball were playing in the Negro Leagues, Josh Gibson and Satchel Paige. So it's in that spirit that I want to talk about this discovery from a team of researchers in Germany who discovered that the sequence of genes on chromosomes, on bacterial chromosomes.
We're not talking the pants that they wear either.
No, we're not talking about the pants. We're talking about the information stored in DNA. But the sequence of genes along bacterial chromosomes have to be precise. That sequencing has to be precise, and it correlates with the functional role that the protein encoded by the gene plays. And this allows for gene expression that is right on time.
Right on time.
For that's the connection. Right on time for the biochemical needs of the bacteria. And so this is, a remarkable discovery because it's completely counterintuitive from an evolutionary standpoint. This is essentially a failed prediction because most biologists would argue that the sequencing of genes in a bacterial chromosome should be random relative to function. There shouldn't be a precise sequence that is necessary. But it also, to me, highlights, once again, just the elegant evidence for design that we see in biochemical systems. And I've spent 25 years writing articles about different facets of biochemistry that point to design, and this is just one in a drop of a very big bucket full of discoveries. But this, to me is really significant because, it reasons to believe we take the view that there's a creator, and that creator is the God of the Bible, and that if indeed there's a creator responsible for the universe and for life, that we would see design in the universe, we would see design in the earth, the earth system, we would see design in living systems, and in fact, we see that. And for the sake of the gospel, being able to establish with some high degree of confidence that there is a creator is necessary for the gospel. I wasn't a Christian when I was in college, and I had friends who were Christians, and they would share the gospel with me. And I just didn't have any need to respond to it because I wasn't convinced there was a creator. It was only later in graduate school, because of the elegant design of biochemical systems, that I became convinced there had to be a creator. Because of biochemistry.
The discovery of design was kind of like a crucial step to get you started.
Yes, and then once I'm convinced there's a creator, now I'm asking other questions. Who is this creator? Do I relate to that creator? So then when I was presented the gospel, it was a very different framework in which I was able to respond to it. So this is really critical to be able to establish design in the universe, design in living systems. And so it's towards that end that I want to talk about this paper, and here's a shot of the paper. This was published in the latest issue of Science, or one of the more recent issues of Science, again by a research team from Germany. And for those people in our audience who aren't up on biochemistry, I'm going to take just a few minutes and give a very quick flurry of background information so people can appreciate this.
So this is a quickie course?
This is a quickie course in biochemistry. And it all starts with this slide here that's showing a picture of DNA, right? And because the bacterial chromosomes are made up of DNA, DNA stores information that the cell's machinery needs to build proteins, which are the workhorse molecules of life. And so if it wasn't for proteins, life isn't going to be possible. And there's got to be information that directs the production of these proteins. And so DNA consists of the well-known double helix that, if you unraveled it, would consist of two molecular strands, and those molecular strands interact to form like a ladder-like architecture that then you twist to get the double helix. You twist in a right-handed way. The strands are made up of these genetic letters, nucleotides, abbreviated A, G, C, and T. That's the information. The sequence of the nucleotides is the information needed to make the proteins. And a region of the DNA molecule that contains the information to make a protein chain is called a gene, right? And so it turns out that, again, the genes have to be precisely sequenced according to the function that those proteins play in the metabolic process. Now, part of what we need to understand, too, is how DNA replicates. Because when a mother cell divides into two daughter cells, those daughter cells each have copies of the bacterial genome, right, or the bacterial chromosome or the bacterial DNA. And that's the way in which each of those daughter cells has the information needed to make the proteins it needs to survive. And so this requires that the DNA is replicated. And the process of DNA replication could be conceptually understood as the two DNA strands separating.
Right.
And we got a slide showing that. And then each of those strands serves as a template to assemble a daughter strand. And so you wind up with two daughter DNA molecules where each one has a strand from the original molecule and a newly synthesized strand. And so for those people keeping score at home, this is referred to as template-directed semi-conservative DNA replication. Now, when this next slide shows what's called the replication bubble. When DNA replicates, even though conceptually we can think of the two strands separating, what actually happens is only a small portion of the double helix unwinds, exposing two single strands in the process, and then those daughter strands are assembled. And there's a precise location where replication begins in chromosomes, and this is called the origin of replication. And the replication goes in both directions at the same time, so you get this replication bubble. And the bubble gets bigger and bigger and bigger.
So I imagine there's some kind of molecular machine that basically runs along the strand and constructs it.
Oh, there's a whole ensemble of machines that are unwinding the DNA, stabilizing the single strands, initiating the synthesis process, and then executing the
manufacture.
Yeah, so there's probably 30 or 40 enzymes that are involved.
And we're talking 30 or 40 machines.
Yes, yeah.
They're basically working like a factory to make this replication happen.
Yeah, there's a lot of similarities between...
I assume you got a name for every one of those, right?
Yes, I do,
Okay.
But I'm not going to go into that.
All right.
But now in bacteria, and this is the next slide I want to show, in bacteria, the chromosome is actually is circular, where the two ends of the DNA attach to form a circle of DNA. But again, it's not a random structure. There's an origin of replication and a terminus of replication. So there's a polarity that is very precise in the bacterial chromosome. And the origin is where the replication begins. And this slide shows the formation of the replication bubble. And what you end up having is a circle within a circle as replication starts. And the circle within the circle gets larger and larger and larger until you reach the terminus of replication, and then the two circles separate into the daughter strands. And just as a historical note, scientists who were studying DNA replication using electron microscopes actually imaged these different replication circles, and that's how they, in part, figured out how DNA replication transpired, is trying to create scenarios that would match what they observed with electron micrographs. But anyway, so that's the background that we need. And so one of the things that the researchers point out is that when DNA replication takes place, you have a phenomena called dose-dependent gene expression. So gene expression just simply refers to the process by which the information in DNA is read to make proteins. That's called gene expression. And what they mean by dose-dependent is that when you start replicating at the origin and going away from the origin, those genes that are close to the origin of replication are going to actually have two copies, which means there's going to be twice the level of gene expression for those genes, whereas the genes that are closer to the terminus are only going to have one copy during the replication process and are only going to be able to then have one copy expressed. So the idea is that genes that are closer to the origin will be expressed at a higher level during DNA replication than genes at the terminus. And so they actually have an interesting diagram in the paper that illustrates this point, where they divide the chromosome into three regions: the origin, the mid-region, and the terminus. And they basically show how when DNA replication always takes place at the same speed, but when the cell growth is rapid, that means that DNA replication occupies a greater proportion of the cell cycle. So when cells divide, they begin a cell cycle where they go through discrete stages, and each of those stages, different metabolic events are happening. But, one of those events that has to happen is DNA has to replicate to prepare for the next round of cell division. For cells that grow real slowly, DNA replication it will proceed initially, but it occupies a relatively small part of the overall cell cycle. So this dose-dependent gene expression is not really very important there. But for cells that replicate rapidly,
It is.
it is.
Yeah.
And so this dose-dependent replication occupies a greater proportion of the cell cycle and is prominent in fast-growing cells. And so what they ended up showing, they looked at 910 bacterial species and a whole ensemble of genes in these genomes, and showed that for about 65% of the genes, they saw a precise relationship between the location of the gene and its metabolic role, where genes that were playing a critical role in essentially cell growth, these would be genes involved in transcription and translation, which is basically the production of proteins, clustered near the origin, and genes involved in motility clustered near the terminus.
Mm-hmm.
And then you had most of the metabolic genes kind of in the middle, right? And so they saw this really strong dependence where it was more important for fast-growing cells than for slow-growing cells. So it's this really elegant design in terms of the sequencing of genes in bacterial chromosomes, and this is not what you would expect from an evolutionary perspective, but it's the type of feature I would expect to see in biochemistry if indeed it was designed by a creator. That degree of ingenuity, and there's an elegance and a sophistication and an orderliness that suggests the work of a creator. But this is a quote from a Science Daily article that was describing this work. And the quote says this: "Are genes distributed randomly along the bacterial chromosome as if scattered from a salt shaker? This opinion, which is held by a majority of researchers, has now been disputed by a team of bioinformaticians, from Germany."
Well, it seems like these Germans figured out that there's three categories of genes.
Mm-hmm.
One category needs to be near the origin. One
Right.
can be in the middle.
Right.
One can be at the end. I'm presuming, Fuzz, that if you were to do a deeper study, we would find it's even more complicated than that.
Right. So this is a very crude
Yeah.
dissection of the bacterial chromosome. I would expect, as you'd point out, that the more refined it is, the more we're going to see a greater degree of ordering.
So this is kind of like a jump-off point.
Yes.
We have found that, hey, there needs to be an order in which these genes appear along the chromosome, at least to this degree.
Yeah.
And so this would be a stimulus to say, hey, we need to actually dig into this more.
Right.
And I also see this as an idea, and why wasn't this done years ago?
Well, it's just you need massive computation power to do it.
Okay.
Because you're looking, I mean, most bacterial genomes are going to have between, probably 2,000 to 4,000 genes, depending on the bacterial species. And to establish enough confidence that this is actually due to some kind of inherent biochemical phenomena as opposed to shared ancestry, you've got to do a large number of bacterial species
So
across
for a few decades, the assumption has been, hey, the order doesn't matter.
Yep.
But maybe that assumption was just developed because there's no way we could test it otherwise.
Well, there was no way they could test it, but it was assumed to be random because that's endemic in the evolutionary model.
Right.
Right. But, from an evolutionary perspective, which is how these researchers studied this, if you have bacteria that share ancestry, you would expect there to be similarities in the sequencing of genes along the chromosome.
Mm-hmm.
That would reflect common ancestry. So you not only have to choose a large number of bacteria, but you have to go across the phylogenetic tree to ensure you're not seeing some kind of effect due to common ancestry, but it's, again, an endemic biochemical phenomena. So you have to use a large number of samples and a large number of genes. So that was part of the reason why this hasn't been done before is
just a computational challenge.
Right, and actually having access to databases of the information that you need.
So how optimistic are you that this is going to be a significant jump-off point that's going to lead to deeper studies?
Highly optimistic.
So you think the funding will be available, the computing power will be available?
Yeah.
And so we can look forward to you coming back here and saying, hey, this baby step has now led to as another step, and we're actually seeing, because I would suspect from a biblical Christian perspective, there's going to be even more complex ordering required to make life function.
Yes.
And it's interesting, we're now at a time where we can actually affirm or put that to the test.
Yeah. Now, something else that's really interesting about this study is for many, when people sequence the bacterial chromosomes, when they sequence the bacterial genomes, they'll identify, again, typically 2,000 to 4,000 genes, depending on the species. Typically, a large proportion of them are genes that they don't know what the function is. So, and the thought is this is actually now a useful tool to begin to dissect function for unknown genes. If depending on where it's located in the chromosome,
depending on its location, right?
That gives a clue now as to what it may be doing, or at least this gene must be playing a role in rapid growth, right? Or this gene maybe is playing a role in normal metabolism in the cell, or this gene may be devoted to systems that aren't necessary for growth but may play other roles in survivability. So interesting how this insight has, broad-ranging ramifications.
Well, I think it's got ramifications for genetic engineering.
Mm-hmm.
If you're going to take, say a chromosome and take out a defective gene and replace it, where you put it is going to be important.
Yep.
And so this may actually make genetic engineering a lot more efficient in the future.
Yeah. Yeah, I mean, that's one of the big problems that researchers face when they're trying to create synthetic cells or use bacteria and genetically engineer them is a lot of times you don't get the results that you expect by inserting genes or even collections of genes. And so you're spot on, Hugh. It's very insightful that, where you put that gene.
Where you put that gene makes a big difference.
Is going to make a big difference. So there's a lot of implications for this. But, to me, we've talked about the origin of life. This also has implications for the origin of life because bacteria are relatively simple organisms. And this is just adding to the complexity of bacterial systems. It's, at one point people were just satisfied trying to know what is the minimum number of genes that you need for an organism to live, independently in the environment. And the number varies from about 1,300 to 2,000, depending on whether it's a heterotroph, a chemoautotroph, or a photoautotroph.
Right.
Then people have discovered that, well, it's not only just having the right proteins, but those proteins have to be targeted to precise locations in the bacterial cell. So you can't just have proteins randomly scattered in the cell. They have to cluster either near the pole, the mid-region, or something like that. So there's an internal structure. That is another dimension of complexity. And now we see even a third dimension of complexity where the information that makes those proteins has to also be precisely organized along the chromosome. How you achieve all of that integrated complexity simultaneously, in an original life scenario is mind-boggling, particularly when we have good evidence that life appeared very early in Earth's history, and it was what the very first cells were metabolically complex. What we're learning about what LUCA would have been like is that this was an incredibly complex cell, far more complex than anybody would have even imagined.
And LUCA is?
The last universal common ancestor.
Yeah,
so it's from an evolutionary perspective, this would have been the cell.
The first life.
That would have, well, it's not, it would be later appearing than first life in an evolutionary model, but it would be very early in life's history, and it would anchor the evolutionary tree. So all organisms would descend from LUCA. And
so Fuz, question, when are you going to come up with an article that puts all this stuff out there and adds to our origin of life model?
Well, there's articles scattered all over the place that bring up facets of this, but the point is that this just really makes some kind of evolutionary origin of life process even more difficult.
Right.
Because it's not just simply getting the right environmental conditions.
Well, that's the article I want to see.
Yeah. So I think this dovetails nicely with what you discussed, right? Is that there are these very stringent set of requirements for the habitat for the origin of life, and then on top of that, there are very strict requirements that you need for molecules to form and then assemble into the very first cells.
So more evidence for God, right?
Yes.
Okay.
Yeah, and where you're seeing this evidence is in biochemical systems, which to me is significant because these are the basic fundamental systems of life. If you're going to see design, you're going to see design there if indeed there's a creator, and all indications are life must come from the handiwork of a creator.
Right. Yeah. Wow, this is really fun, Fuz.
Yeah. Well, thank you, Hugh. All right, well, and thank you for joining us on today's episode of Star Cells and God. Please join the discussion in the comments below. We all love to read your thoughts and your perspectives on the discoveries that we share. Also, remember to like this video and to subscribe for more content. New episodes of Star Cells and God release every Wednesday and are available here on our YouTube channel and on your favorite podcast app. Be sure to share this video with a friend, and remember, the more we know about science, the more we have reasons to believe in the triune God of the Bible.